So without further delay, it is my pleasure to introduce our first speaker, Dr. Maryann Martone, Professor Emeritus in the Department of Neurosciences at the University of California at San Diego, and she is the Principal Investigator of the Neuroscience Information Framework and National Institute of Diabetes and Digestive and Kidney Diseases Information Network. She is the principal investigator of SPARC's Knowledge Management and Curation Core, K-Core as it is known, which is one of the 4 cores that comprise the SPARC Data and Resource Center. And she has been leading the overall SPARC curation effort since 2018. She is an international leader in the field of neuroinformatics, fair scholarly communications, and open science. And today, Dr. Martone will discuss the SPARC Portal and Knowledge Base. And so we'll go ahead and have Dr. Martone join us, and we'll be introducing Dr. Peter Hunter, who will be our second speaker, and Dr. Sue Tappan will lead the Q&A session. Okay, Dr. Martone.
Thank you, Patricia, and thank you very much for inviting me. I hope my slides have very rapidly come up on the platform.
Yes.
Excellent. So thank you very much, all of you, for joining and for participating in our poll. We are going to, myself and Dr. Hunter, introduce you to some of the SPARC tools for integrative physiological modeling. For those of you who are familiar with SPARC, we hope to show you some new features that are available. And for those of you who are not familiar with SPARC, we hope to introduce you to the portal and all that it can offer. So the first part of the presentation, which I will give, I've entitled A Tour Through the SPARC Portal. So we'll start with what is SPARC. And what is SPARC? If you see where that arrow is, SPARC is a open neuroscience and systems physiology platform that contains multi-species data, knowledge, computational modeling, and spatial mapping. So one of the first things that is new is that we have a new tagline. SPARC is responsible for bridging the body and the brain, and we really are focused on the peripheral nervous system and its CNS end organ interactions. But we are also an open data platform where you can share your data and models in support of the aims of SPARC to develop treatment that will change lives. So if we look at what SPARC offers, or what you can do with SPARC, there are basically 4 different pieces of the SPARC platform. First is you can browse, view, and get data and models. You can view 2D and 3D anatomical and functional maps, create computational pipelines, and also use SPARC to fulfill new data sharing mandates and support open science through the sharing of data. I believe a few weeks ago, Dr. Ezra Newfield from SPARC presented the SPARC computational platform. And so here we're going to really be focusing on the data and models, how you use the SPARC portal. And then Dr. Hunter will talk about the powerful 2D and 3D maps that are available. And I'll also talk a little bit at the end of my presentation about the data sharing platform and how you can use it in your research. So one of the things that I said was new was the SPARC branding, this bridging the body and the brain. And I thought it would be useful to give a little of the history of SPARC, because many of you may know it under the term stimulating peripheral activity to relieve conditions. That's in fact what the SPARC acronym stands for. And it was originally established via an NIH Common Fund program, which runs through 2025, that was designed to promote the development of more effective and targeted neuromodulation therapies. Neuromodulation therapies, of course, is an emerging therapeutic approach to treat diseases of the autonomic nervous system by providing scientific and technological foundations for future bioelectronic medical devices and protocols. The first phase of SPARC, which started roughly around 2017, had a large consortium that was gathering multimodal and multi-species data, largely about the autonomic nervous system and its end organ CNS interactions. That phase ended in 2022, and phase 2, which is currently underway, is focused on obtaining very detailed anatomical and physiological data sets about the human vagus. These are going to be very large, important data sets that are going to start coming online in the next year or two. But in 2023, SPARC began to look to the future as the SPARC program was winding down. And we have become an open data repository that can be used for the larger community that is dealing not just with the ANS, but the peripheral nervous system, body-brain interactions in general. So this is an exciting time, and if you'll start to see in SPARC, we are changing our branding over time, but you still will see remnants of the existing SPARC project in there, which is why I thought it was important to introduce it. And over here on the right, I just want to point out that already some of the data that has been generated by SPARC Phase 1 is starting to be used by the scientific community at large in order to further the aims of bioelectronic medicine. So how do you get started with SPARC? The best place to start is to go to the SPARC Portal, which is available at sparc.science. This gives you the overall navigation of the many different pieces of the portal that are available to you. And I want to, even though I'm going to focus on the data and models and later a little bit on maps, also highlight that SPARC really is a community hub to bring together the integrative physiology and anatomical communities. There hasn't been a large effort that has been devoted to this community. So we also focus on things like news and events and community highlights and spotlights to help the community stay in touch with each other and the exciting developments that are going on. But of course, usually people come to us and they want to find out how to find data and models or do something in specific. So I thought I would go a little bit through the data search. There are many ways to start. You can see here we have find data by category. So we've highlighted by pre-faceting some of the searches so that you can search for data and models around these different systems. But another way of accessing it is by clicking on this data and models link at the top. When you do that, you will be given a list of data that are available. There are currently over 250 data sets and models available through SPARC. You can see at the top, we've divided these up into different types of categories: data sets, anatomical models, computational models, but we also feature the projects that SPARC supports so that you can get a good idea of the community that's working in it. Here we've taken advantage of the faceted search that SPARC allows. You can see some of the facets down here, which will allow you to select different types of data. So if you have good eyes, you can see that I've selected the electrophysiology, physiology, and mouse facets right here. But there's also a keyword search so that you are not limited by what we provide over here. You'll get a listing of the data sets over to the right. Here we have 24 results that you can see. And you can also go through the different categories up here to see what we have in those. I'm going to focus on the data sets here. So if you click on one of these data sets over here, this one is called optogenetic activation of nitrogen and cholinergic neurons of the marine colonic myenteric plexus. All SPARC data sets undergo semi-automated and human curation. So SPARC is highly curated so that all data sets are organized in exactly the same way to make it much easier for you to browse and understand a given SPARC data set. Because we are multimodal and multi-species, nevertheless, they are all organized in exactly the same way. And as you saw, they're annotated with a common vocabulary to make it easy for you to use these facets in order to be able to find them. For each data set, you see this top tabbed interface. This tells you different details about the data set. So the abstract here gives you an overall description. All of our data sets are linked to experimental protocols that have been deposited in protocols.io. This is both to give the experimental context that is necessary to understand a given data set, but it also provides a very good way for researchers to highlight and share their protocols with the community through the protocols.io platform. And I should note that SPARC protocols, the ones that are there associated with SPARC data sets, receive well over close to 40,000 different views. So this is something the community very much cares about. I'm going to go through some of these other tabs up here, starting, of course, with the files, because you would like to be able to see what data sets are available. There are ways to download or access data in the cloud. There's also a browsing mode where you can look at SPARC and some basic tools that allow you to download individual files if you wish. But you can see here that there's 3 categories: derivative, primary, protocol. There's some top-level file folders here. The primary folder is the one that has the data in a form which researchers are most likely to find useful. So it's not completely raw, but it is also minimally processed. And as I mentioned, all SPARC data is curated to the same standard. So if you click on that folder, you will get a list of folders that are named according to a subject. And in order to understand what that subject is, you need to go over to the subject's spreadsheet here that contains all of the demographics. So we provide a tutorial up here that allows you to navigate a SPARC data set, essentially go through this hierarchical structure and match the subject's samples and others files with their metadata. But because SPARC datasets can be very complicated, and trying to browse by going through this hierarchical browsing mode can, in fact, lead you down very deep, and then you have to keep coming back. It's very difficult sometimes to overall understand how a dataset is organized. We've recently introduced a new tool that's available up in this menu here called the SDS Viewer. If you click on the SDS Viewer, it takes you to a third-party tool that was developed by a company called Metacell that essentially displays the entire data set as a graph and provides the metadata in a side panel. So at every step of browsing this particular graph, you can see the metadata that's associated with individual subjects and samples and data sets. So it's basically an integrated way to view the overall structure of the data set. If you select, for example, an individual subject, it gathers together the primary and derivative data. You're not meant to read these things, but you can see at the endpoint, you get the individual files. Over here on the right, it tells you that the subject is a female and how old it is and what subject group it belongs to and its species. And you'll notice that in the left panel, you still have that hierarchical navigation so that you can see what this looks like on the SPARC portal. So this is a very, very powerful tool. We're adding more functionality all the time. It's available in beta form, and we welcome you to play with it and give us your feedback. SPARC, as you may have seen that tab at the top, there's something called CITE, C-I-T-E, because SPARC supports credit and attribution for those who are sharing their data through SPARC. We provide full data citation support so that you can export data citations for an individual data set into your favorite reference manual, reference manager for EndNote, et cetera. And we're seeing people, we encourage people and give them instructions on how to cite SPARC data so that you will get a citation count for your data set, just like you get for your papers. We also track things like downloads so that you can measure the impact of data sets that you may have shared on SPARC. To explore SPARC data online, as I mentioned, you can download files. You can also download the entire or access the entire data set in the cloud, but we also provide a lot of online tools to allow you to explore the contents of data sets without having to go through an additional step. So in this case, you access the Gallery tab. The Gallery tab will provide a set of panels that will tell you the types of tools that are available for that particular data set. In this case, it's a physiological data set, and so there are tools that will plot the contents of the data files so that you can get an overall view of what the contents of individual files are. And of course, if there are imaging data, we have online viewers, including the BioLucida image server, so that you can explore 3D stacks and 3D visualizations all from your web browser. But to lead us into the second part of the talk, you'll also see over here on the left that there's something called the mouse flat map. So the mouse flat map links data sets inside of SPARC into the map portion of SPARC, which provides detailed visualizations of the spatial organization and functional organization of connectivity through something called SPARC flat maps. So if I click on this, I am taken to one of these flat maps. In this case, it's a mouse flat map. We have them for multiple species. You see that the colon is outlined here so that you can see whatever organs you're looking at in context. But if you further click, you see this cabling here, which represents connectivity, largely of the ANS at this point, reflecting SPARC's origin, but being expanded into general sensory and motor functions as well. These are very detailed routings of individual connections that pass through nerves. And Peter is going to talk a lot about these, specifically focusing on tools that we have for functional connectivity. But I'd like to show you that, in fact, it is multi-scale. So if you zoom in on these maps, you see detailed circuitry. And if you look up at individual nerves, you see that multiple types of neuron populations actually pass through these. And so where we have nerve information, we can start to tell something about the makeup of individual nerves through these maps. And as I mentioned, Peter is going to talk about this in more detail, but the part that I wanted to talk about is that all of those visualizations, they are not hand-drawn. They are, in fact, derived automatically by Peter and his team from something called SCKAN, which is the SPARC Connectivity Knowledge Base. So this is a detailed knowledge base containing that type of connectivity that you saw, and it has been constructed by consultation with experts who provide their views of how circuits of different organs are put together. The published literature, we have an AI-based NLP tool which scans the literature for connectivity information, and we curate that and we put that into the knowledge base. And also we extract a lot of data from individual SPARC data sets that, of course, many of which have information about the connectivity of individual organs. The knowledge base incorporates a model of the neuron. So it's not just regional connectivity that A goes to B, but we note the locations of cell bodies, axon locations and axon terminal locations, sensory terminal endings, and dendrites. And each connection includes species, sex, synaptic connections, evidence, and associated with it, and also includes measures of uncertainty, which is in development right now. So it's a very, very powerful knowledge base. As I mentioned, this drawing here, this cabling all comes directly from SCKAN, but because we have this in a knowledge base, we can drive all kinds of powerful visualizations that will tell you something about the connectivity of individual neuron populations, or looking overall at things like heat maps of how heavily connected different structures are. And so as you interact with SPARC in the coming months and year, expect to see a lot more of these types of visualizations that are driven by SCKAN. SCKAN, as I mentioned, has the model of the neuron in it, so it enables very powerful queries where you can point to a location in a nerve, for example, and it can tell you which populations of neurons pass through it, whether there's a soma associated with it, an axon terminal. So it really starts to support the idea that if I were to virtually stimulate this, I would know something about the populations of neurons and therefore anatomical structures that were affected. We've just recently released a beta product called SCKAN Explorer. It is a query interface to allow people full access to the contents of SCKAN, which will allow you to do these types of queries. It's a very preliminary tool, largely at this point used by curators, but if you poke around, you can see that for the types of connections that you're looking at, you can query for different pieces here, and you get the species, the sex, and all the references that went into actually creating this. And it is hooked to little visualizations that help pull out some of these connections into a simpler form so that it is easier for you to look at. This is meant to be a community tool, so we are also developing tools that allow the community to contribute knowledge, correct, edit, add knowledge to it. And this is something called SCKAN Composer, which will also be coming out soon, that will allow different groups or individuals to contribute to this major effort. So I just want to end with a plug about the fact that, as I mentioned, SPARC is now an open platform. We are listed on the NIH NLM list of repositories, of approved repositories that meet the general requirements for the NIH data sharing mandate. So we hope that you'll start to think about sharing with SPARC. We can help you with these new data mandates. So just to sort of summarize, SPARC is a very powerful, now well-populated database, open platform that offers this repository for examining and using data sets associated with the PNS. It allows you to curate, manage, archive, and share data, and as you can see, in a very professional way. And those of you who might be a little intimidated when thinking about submitting data to SPARC, one of the things about SPARC is that we have dedicated customer support. We have curators and software tools to help you interact with SPARC and upload data to it. So I think Sue's going to be sharing materials at the end that will give you more information on how you can use SPARC. Thank you.
Wow, I am most impressed with your presentation. Thank you so much, Dr. Martone, for sharing the power of SPARC. I'm not sure that many people are aware of its power, so thank you for that.
You're welcome.
Okay, what I'd like to do now is bring forth our next speaker, and that will be Dr. Peter Hunter. And Peter Hunter is the Distinguished Professor and founder of the Auckland Bioengineering Institute at the University of Auckland. He is the principal investigator of SPARC's Spatial Mapping Core, MapCore is also known as, and that is another core that comprises the SPARC Data and Resource Center. As co-chair of the Physiome Committee of the International Union of Physiological Sciences, he has been helping to lead the project, which aims to develop model and data encoding standards and to use computational methods for understanding the integrated physiological function of the body in terms of the structure and the function of cells, tissues, and proteins. Dr. Hunter will cover scaffolds and the functional connectivity map for physiology and mapping. Welcome, Dr. Hunter. I can see you, and we can share your slides. We'll be ready.
Okay, thank you, Patricia. Hello, everyone. I'm very pleased to talk in the second section of this webinar particularly about how we could be developing the SPARC portal for supporting physiologists, particularly in relation to multi-scale modeling applications, data and modeling. So what I'm going to talk about is particularly the functional and the what we call scaffolds for dealing with spatial. I'll talk about that first, the spatial encoding of information that we're using for various organs in the SPARC project. And these two are two of the three maps that Dr. Martone referred to earlier. So first of all, for scaffolds, for representing spatial fields over, representing spatial data over various organs or at any level really of physiology, we've developed what we call a CAD computational program to be able to represent anatomy, and specifically for anatomical features, which unlike engineering are often extremely smooth surfaces compared to sharp engineering borders. So we've developed a whole suite of computer-aided design tools and primitives that allow us to build more complex organs. So some of them are shown here for octagonal or cylindrical or spherical shapes that can then be, they're ways of mathematically defining spatial regions, and we can build them into quite complex structures, shown more here on the right. Here's a stomach, for example. Here's an element of the colon. And I'll show you how these are used in the next slide. So a typical example would be where you want to be able to represent the shape of the colon, but do it in a way that's applicable to multiple species. So for human, pig, and mouse, in each case, there's a center line is defined, which is obviously very different in the three cases. But once you have that center line, then we can define the haustra, representing the detailed structure of the antennae, coli, etc. For the colon, which again are very different across species, but the same common framework can accommodate any mammalian species. And then we can add the haustra to the center line to generate a colon for the human, pig, or mouse. And that means that we're in a position to compare data sets across different species or within one species, individuals within one species. Same thing for organs like the heart and the lungs. We develop scaffolds that represent the geometry of the heart with subtle variations in terms of the number of pulmonary veins returning to the left atrium, for example, which can be typically 3, 4, or 5. But other than that, the topology is the same. Then we can refine these meshes to export them depending on the resolution that's needed for capturing data or the resolution needed for capturing the physics of computing using computational physics to understand the function of these organs. We can generate the meshes for those computational methods. Here are examples for the lungs, again, with the original scaffold that is applicable to multiple species and can be fitted to any species, but then an example of a more refined mesh that's generated for a particular application. Then, as Maryann mentioned, we've got a number of people using these scaffolds for registering neural data, or in some cases vascular data, into the scaffolds. And this becomes a way of comparing different data sets across either the same individual for a given species or across species or across individuals within a species, because we've always got that common coordinate reference framework defined by the material coordinates of the scaffold in order to be able to consistently reference the same location for subtle differences in the shape. And here's one example of doing that, where showing the advantage where the scaffold, in this case a mouse colon, can be cut along the mesenteric border in the same way that the experimental preparation is cut, and then opened out so that the actual experimental observations of structure, in this case vascular structure, within that tissue sample, we can apply the same forces that you do experimentally to stretch out the tissue to make the microscopy observations on. We can do the same with the scaffolds, but then we can fold it back into the 3D structure to represent the data that's been recorded in the flat preparation. We can demonstrate it or display it rather within the context of the three-dimensional organ. Then we can embed these scaffolds within 3D bodies. So we have a scaffold for the whole body, and we can place the various organ scaffolds within that 3D body and then adjust the shape of that body to meet any individual person or any individual in a given species. So I want to now switch gears a little bit to the functional connectivity FC map, which is the third of those maps available through the SPARC portal, because I think this is a map that's going to offer us the potential to really link with a lot of physiology work. And I'm really looking for feedback from the physiology community into how we should develop these maps. And the idea basically is that we lay out the entire organ systems of the body. Each of these blocks corresponds to an organ system. Here's cardiovascular, for example, and this is neural. And the idea is that the map provides all of the information on the map is coming from the single knowledge base, which Maryann talked about, and can be rendered on the map, or it can be rendered into the 3D body consistently. But what we want to be able to do with the FC maps is to be able to zoom in to particular regions. So each of these little green boxes represents a what we call a functional tissue unit, or a small physiologically relevant functional component. And the idea is that we can then define a lot more physiological detail within each of these regions and zoom in, but make the connections from the small scale up to this big picture of the body, which has all the organ systems. And we can then just display just the information that's of interest for a particular physiological investigation. And remember that all the information displayed here is going through a curation process and coming from that common knowledge base. So the question we're asking is, how can we develop the FC map to help understand and model physiological mechanisms that depend on multiple spatial scales? So one example would be if we wanted to model blood flow at the higher scale. So we have all the blood vessels, the arteries and veins, coupled into all the organ models. And on the left here is shown all the coupling into the muscles of the body. And then we can display that in a three-dimensional context in the 3D body. And then we can solve the physics of pressure and flow around the body. And then we can display that information in the context of the flat map or the 3D body in order to interrogate flow in given locations. These are being beta tested as part of the FC and 3-body maps at the moment. And then once you have information about the pressures and flows around the body, the obvious thing would be to be looking at transport processes, again, all in the context of the FC map, which is zoomable. So you can zoom in like a Google Street Maps as much as you like into the particular areas of interest. So for example, if we were interested, and this is work I'm doing with Bernard de Bono, if we were interested in the transport processes associated with the kidney, liver, and gut, where we can solve for the blood flow that's entering each of these systems, and we can also solve the equations that link the fluid flow within a system. For example, glomerular filtration leading to all the way through to the bladder in a vertical sense here, or in the case of the liver, the bile, in the case of the gut, the chyme, and the flows from the stomach through to the colon. But now we want to be able to link in a particular solute or a drug that's being taken up and dealt with via various aspects of the body. So it may be that, for example, if we look at the kidney first, the glomerular filtration process means that the solute that's in the urine in the flow going down this path now needs to be brought back into, reabsorbed into the tubules of the nephrons of the kidney. So we need to be able to represent the transport processes at the level of the endothelial and epithelial cells that involve the transport routes between an epithelial duct here and the capillary in the peritubular bed. And so that means we've got to be able to deal with the various proteins that deal with transport processes, either intracellular or pericellular, that involve movement of that solute from the epithelial duct through to the capillary, so through this path here. So this is our more zoomed in part of the anatomy that's relevant to the proximal tubule, for example, and we need to be able to move between the whole body of the FC map, but zoom into a particular part of the kidney for the transport processes at this level. Likewise, and of course, we've got to also deal with water reabsorption as part of the flow process. Likewise, we might be dealing with transport processes in the gut, where we're picking up substances from the gut, which will then be likewise dealt with at a protein level, transporter level, within endothelial and epithelial cells in the gut, in order to then look at the uptake into the or either uptake from the blood into this chyme or vice versa. And similarly for the liver, the transport processes associated with liver transport and bile. So all of the protein function represented in those models are dealt with via physics models based on the bond graph concept, which ensure that conservation of mass, conservation of charge, and conservation of energy is always satisfied at all scales. So for example, at the level of the kidney, if we're looking at one particular protein, let's say GLUT2, SLC2A2 protein, what we want to be able to do is represent the various reactions going on that represent the chemistry, in this case of glucose transport from outside to inside. And that involves the protein having an outward facing state that then binds a substrate, in this case glucose, then transitions in the membrane to a state where it's facing the inside of the cell and releasing the substrate and then returning to its original state. So it's cycling through those states, and we're dealing with the reactions involved in binding, unbinding, and forward and reverse states of the protein for this particular simple reaction. And out of all of that, we can get also a reduced model where we're interested primarily in the steady state flux through here. And it turns out that binding and unbinding is very rapid, so we can make that assumption. We can assume a constant enzyme cycling rate, and then we get down to a model, in this case with only 2 parameters, that we can use in much larger scale models. But then when we look at the whole of the SLC family, nearly 500 genes and 66 gene families, which are transporting various small molecules across membranes throughout the body. If we look at the way those families are organized, in fact, the 66 families of those 500 genes all reduce to one bond graph template, depending on whether we cut it off here as a single ligand, a double ligand, a triple, or 4 ligands. So with one bond graph model, we can capture the function of all of those genes. And similarly, another example is metabolic reactions, where we'd have one bond graph model, which in its reduced form with multiple products, multiple reactants binding and multiple products for the reaction, we can then represent with one bond graph model that can then be built into, for example, dealing with all of carbohydrate metabolism here in a very computationally fast manner with these physics-based models. And then what we can do again within the context of the FC map is to automatically couple these models into higher scale structures. So they're sharing GLUT2 and SGLT, for example, are sharing a glucose source both on the outside of the cell and the inside. Likewise, for other transporters, would be sharing sodium. And so we've got the ability to automate the process of combining these transporters and then begin to build higher level structures. So for example, in the kidney, these are the SLC proteins involved, or at least some of them, and then also involving ATPase, ion channels, and aquaporins. So we can begin to automatically build all the small units that govern the function of recovery from the tubule to the capillaries in the proximal tubule on an automated basis using these models that are always adhering to the physics of mass, charge, energy conservation, and then that guarantees that the units that are built also satisfy those conditions. So in summary, what we're wanting to be able to do is to use a single knowledge base, SCKAN, as Maryann has described, use the FC map to render annotated physiological components and connectivity. And that connectivity could be neural, could be vascular, could be lymphatics, etc. And then we want to be able to capture spatial information where it's relevant at all scales with the scaffolds. So in parallel with the FC map are the 3D body models with their organ scaffolds, but scaffolds at any scale, then with the ability to zoom into any component of the map to access the detail, and the ability to only display just the information that's relevant to that particular study, physiological study. And then, as I've mentioned, the ability to use an energy-based multi-scale framework for models at all scales. Just like to acknowledge the people at the SPARC team at the University of Auckland who are contributing to this, and thank you.
Great. Thank you so much, Dr. Hunter. SPARC is very, very detailed and intricate, and I'm really looking forward to hearing the feedback that we receive when we get to our next poll, which is shortly coming up. So thank you for your awesome presentation. And what I'd like to do before we begin our Q&A is launch the poll that you now see on your screens. If you could take a moment to respond. And looking at the feedback, wow, we're almost tied with everything. Maps and connectivity leads with 38%, followed by data and models, and then computational tools. And before we start the Q&A, Dr. Hunter and Dr. Martone, did you want to comment on any of the poll results that you've seen?
No, I think that's probably very consistent with the way in SPARC we're trying to develop all three of those in parallel, because they do all interact.
Yeah. What about you, Dr. Martone?
Yes, I agree. One of the reasons that we spent a lot of time building the knowledge infrastructure SCKAN is that in SPARC, we really tried to make sure that there was a common framework that underlay all of the different pieces. So we have, powerful ontologies, knowledge graphs, and other things, and they percolate through the different parts of SPARC so that it makes it easier for them to be able to be used independently, but that underlaying that, they all add to each other and they're all heavily interlinked. So the anatomical maps connect, or, the data connects to the maps, which connects to the knowledge base, which connects to, some of the modeling capabilities. So we're not completely there yet, but the foundations really have been laid.
Okay, well, great. I want to thank everyone for their feedback. And we're going to start the Q&A session that will be led by Dr. Susan Tappan. And she directs the efforts to expand and support the growing SPARC community. But this does not even begin to explain who she is and what she does behind the scenes. And before we began our session today, I understand that she's a networking guru. And I would just like to give her the just introduction that she deserves. So Dr. Martone, can you start off by introducing us to Dr. Tappan, and then I'll let you say a few words about her to Dr. Hunter. Let's just give her the introduction she deserves.
Well, I think that Sue is responsible for us being here today because she has a couple of hats at SPARC. One is basically community outreach, community liaison, and she established a relationship with APS. SPARC has been, of course, active at the last couple of conferences. But she also works with me on the knowledge base aspects, the composer tools, the community tools. Come via Sue. She has a deep background in microscopic imaging. Imaging. She's an expert on image formats, microscopy, 3D microscopy. So she's a extremely valuable asset to SPARC. And as she's come on board with communications, I would say that our outreach really has exploded.
Great. Dr. Hunter?
Just to reiterate, Sue is wonderful to work with, and we're very dependent on her ability to connect people right across to other communities outside SPARC as well as within SPARC. Much appreciated.
Okay, well, with that said, Dr. Tappan, I'm going to turn the moderating of the Q&A session over to your capable hands. Thank you, and the speakers will stay present.
Thank you very much, Patricia. I want to just thank you both for such a great presentation. I think that SPARC benefits dramatically from your combined vision. And one of the things that I'd like to pose to you first, a question that has come in is, Peter, when you mentioned multi-scale modeling, how are you dealing or planning to deal with the tissue function level?
What we're doing is trying a number of different approaches, and it does differ depending on the organ of interest. The kidney is a rather lovely example where you have most of the function distributed in a one-dimensional line along the tubule and the collecting ducts. And so we can deal with transport processes along, spatially dependent along that single line, but fairly readily be able to construct models that initially target specific mechanisms within cells, but then build those into the whole nephron and then scale up. So the role of the scaffold then is to define all the the ratios of area to volume and length, etc., throughout the anatomy of the kidney, and to provide a spatial context for altering the parameters appropriate to their position within that spatial context. But in other organs, it's very different. I mean, in a sense, a kidney is well represented by the function of its nephron, and the whole kidney is just scaling that up to 1 million nephrons typically for a human, or 100,000 for a mouse, etc. But in the case of the heart, it's rather different. The heart its function really only emerges at the level of the whole organ, where it's acting as a pump. And so very much the tissue there, we need to define what we call constitutive laws that capture the relationship between stress and strain, and how it depends on subcellular function. But then that plugs into higher order, what are called finite element models, where you're solving partial differential equations for the function of that very integrated organ. So each organ has its own challenges for how we build from cell to whole organ function. But what is important is that they all use a common energy-based framework for making sure that we're satisfying the laws of physics at every stage in that pipeline.
Thank you very much, Peter. I love that even at such a deep level, the concept of bridging the body and the brain is really factored in even at this subcellular level that you're talking about. The components of how data is maintained on the SPARC portal, I think also allows that to be integrated into SCKAN, right? So we're using information from the literature and also information about the data that is contributed into the SPARC portal to refine the knowledge graph. Is that correct, Maryann?
Yes, that is correct. The knowledge graph itself, as I mentioned, it comes from multiple sources. So it is really meant to bridge the data sets knowledge that people have about it so that it can be exposed to query and visualization so that we can vet whether it is correct or not. One of the reasons that SPARC was initiated was that in many cases, our maps of peripheral connectivity are based on textbooks that are 100 years old. And of course, errors get in there, unsourced things get in there. So the idea of SPARC is that we expose not just the new data that is coming out, but also place that in the context of what was already known so that we can see where the gaps are and where inconsistencies are. So the same vocabularies that are used to annotate our data sets are used to annotate connections, are used to annotate the 3D scaffolds that Peter showed, and the models, so that there is this very tight integration underlying all of SPARC products.
Thank you, Maryann. I have a question that I hope you will each answer, if you don't mind. The question is, what's next for SPARC? How do you see it expanding into or across fields? Peter, you want to start?
Well, what I would like to see is that the framework that's been built up over the last 7 years, which primarily had a neuroscience focus, obviously, but is now expanded to include, particularly initially, I would say, the circulatory system. So cardiovascular and respiratory, and the sort of transport processes and questions where you need to bridge scales, but in the context of understanding blood flow around the body and flow of other fluids as well, as I indicated. That to me would be the most obvious first step extension of SPARC into other areas of physiology. But ultimately it could expand into all areas of physiology, the same concepts where you're treating the body as an integrated whole, but you're also recognizing that so much of high-level physiological function is dependent on what proteins are doing. And in a medical context, of course, you're using blood biomarkers to tell you about function at the protein level, as well as imaging and whole body physiological testing to understand function at the whole body level. So we've got to connect those together. We've got to be able to integrate the body as a whole, all the organ systems of the body, but integrate them in a multi-scale sense that really brings in the physiology at all levels, from proteins right up to the whole body, but in a way that uses physics, because all of these mechanisms have to obey the laws of physics. So that provides a great simplification to how we interpret physiological data.
Simplification. I love that. It doesn't seem simple to me, but I appreciate your vision on that topic. Maryann, how would you, what do you think is sort of where you see SPARC going next? What's next?
Well, I think SPARC, in my view, fulfills a very specific role right now, in that it really serves as an aggregation point and a community focus point for research that concerns that interaction between body and, as we say, brain, right? The central nervous system. We know that funding at NIH and others is very heavily brain focused. In the brain, it's very heavily cortex focused. And of course, the types of massive data gathering exercises we can do now allows us now to connect up the rest of these parts. And so we knew in SPARC that we needed to expand beyond that limited focus of the ANS, because of course there's a lot more going on in the periphery. But I think where SPARC goes, given that it does have this community hub, I think, focus, and would like to have this community hub focus to sort of bring together all the communities that work in this area, we'd like to see what the community needs, right? And be able to let that direct where we're going. So one of the things that SPARC is assembling is as an open platform for data and tool sharing is an advisory board that represents different parts of the community that can help us, help guide us as to where we need to go. There are opportunities, but of course, as one expands, there are usually way more opportunities than there are resources to meet them. So, we are balancing, like every other project, the fact that unlike our institutes of health and unlike our anatomy textbooks and everything else, the body is not neatly parcellated into anything, and there's not neat divisions between scales. But at some point, of course, you have to say, this is the part that we're going to focus on, and this is our expertise. The beauty of the fact that more and more people are paying attention to fair data principles, that things need to be interoperable and reusable, means that we are building things in a way that we can integrate with other resources as well. So we don't have to be everything, but we should do our part really well, and then make sure that we can integrate with other projects that are coming online or already exist. So I would say that from my perspective, this is our focus, but I think we are very responsive to the needs of this community to have places to share this data and tools and the types of powerful applications, as you saw with Peter's work, of what we can do with this once you get everything in this common framework. It's very, very powerful.
Last question before I toss it back to Patricia is the SPARC Common Fund program the only consortia that is depositing data to the SPARC portal?
No, in fact, we have two other major consortia that are depositing data in SPARC from the HEAL program. The Rejoin HEAL, which is focusing on the anatomy and physiology of the knee and the temporomandibular joint, the TMJ. Of course, the HEAL program itself is concerned with pain and being able to do the basic science that will help us solve things like the opioid crisis. And then SPARC is also providing the platform for Precision HEAL, which is looking at the dorsal root ganglia and the genetic, molecular, and anatomical, physiological makeup of the DRGs. So there are at least two major consortia that are currently using SPARC as their platform.
Excellent. Well,
and we
thank
there will
you.
Be more. Yes.
Yes, we hope there will be more. And you don't have to belong to a consortia in order to contribute data. We are seeking data and models and computational simulations. We invite you to utilize the tools that we have available on the portal itself, whether you'd like to map your spatially segmented image data to a three-dimensional scaffold of an organ system that we already have in place. We'd like your data. We'd like you to help us expand our community and build the body as Peter has represented in his 3D body scaffolds. So we look forward to working with you. And Patricia, I will pass it back to you.
Well, thank you so much, Dr. Tappan, for moderating the Q&A. And if you want to learn more information, I did place some email addresses and their website in the chat, so feel free to use those links. And we are about out of time, but before you depart, I want to make sure that you take part in the post-webinar survey. We would really appreciate your feedback. And I tell you, this was a great, wonderful webinar, and I want to thank you once again, Dr. Maryann Martone, Dr. Peter Hunter, and Dr. Susan Tappan for all of the contributions you made to make this webinar a success. And most importantly, I want to thank you, our attendees, for your time and for joining us today. Thank you.
We'll see you at the APS Summit in April.
Yes, April 4th through 7th. So make sure everyone sign up and register. It opened yesterday at December 11th. Bye-bye.
Bye.
Bye-bye.